So today we are going to have our first talk from Georgia Isom from the Eckert and Bhabha lab. And just on a couple of housekeeping notes for the seminar series, we're planning to go through the end of July. Those talks will be posted on the Lipid Research Division at ASBMB in probably the next week or so. We're not totally sure what our plan is going forward in August, but we'll definitely be here through end of July. And so thanks again, everyone, for participating, and we're going to be recording these and posting them there as well. And so with that, I will introduce Damien Bhabha-Ekiert, who will be introducing Georgia. Take it away, Damien.
Thanks. Yeah, so it's my pleasure to introduce my postdoc, Georgia Isom. As you'll hear as she starts speaking, Georgia is from the UK. So she did her undergrad at the University of Bath, finished that up in 2012, and then did her PhD at the University of Birmingham with Ian Henderson. And I think this is where she first fell in love with MCE proteins, which is what she'll be telling you a bit more about today. So during her PhD, she used biochemical and genetic approaches to study this family of transporters called MCE in E. coli and Salmonella. And then in 2017, she moved to New York City to join the Bhabha and Eckert joint labs at NYU School of Medicine. And so we started our labs in 2017 as well, and Georgia was our first postdoc. And so we were really thrilled to have her join our lab, and she's really been a pleasure to have in the lab. So she's done some really fantastic work and contributed to many different projects, and has really been focusing on trying to unravel the molecular mechanisms that underlie lipid transport by this MCE transporter family. So Georgia has been awarded a postdoctoral fellowship from the American Heart Association, which currently supports her research. And she's had several great papers and preprints over the last few years. So she's beginning to prepare for her job hunt, looking for an independent position in academia back in England. So if you're from one of these institutions, please remember her name, Georgia Isom, because I think you'll want to try to snap her up. She's really fantastic. So today she'll be telling you about a joint project she undertook with Nicola, who's a scientist in our lab. And so with that, I turn it over to you, Georgia. Take it away.
Okay, thank you, Damien, for that introduction. And everyone, you can see the slide, correct? Yeah. And thank you very much for John and Mike for giving me the chance to present this. I'm excited to present this to this Lipid Seminar Series. So yes, today I'm going to talk to you about this envelope spanning tunnel and how we think it's involved in lipid transport for the bacterial outer membrane. And just to start off with, as Damien said, this is a joint project with Nicola, and really he was really a great person to have led this project with, and the paper would not exist without his contribution. Okay. So just to start off with, I'll remind you of what the gram-negative bacterial cell envelope looks like. It has an inner membrane made of phospholipid, an outer membrane that's an asymmetric bilayer with phospholipid in the inner leaflet and lipid polysaccharide in the outer leaflet. Now, between these two membranes is the aqueous environment that is the periplasm, and this houses the peptidoglycan cell wall. So the presence of this outer membrane is important because this actually protects the bacteria from horrible substances in the environment, and that includes things like antibiotics. And therefore, bacteria that have an outer membrane are inherently more resistant to antibiotic treatment, and therefore are more challenging to treat. And this is simply because this acts as a physical barrier to protect from these drugs. So when building the outer membrane, the bacteria have the challenge of transporting hydrophobic substances across this aqueous environment. And we know quite well how LPS gets to the outer membrane. However, we're still not entirely sure how phospholipids get across the space. So that is something that we are interested in studying in our lab, is how phospholipids are transported across this periplasm. And today I will talk to you about a class of proteins called MCE proteins. And they're called MCE proteins because they contain an MC domain. And I'm just going to start off by giving you some pieces of evidence that these are candidates for phospholipid transport for the bacterial outer membrane. First of all, these proteins are really widespread across bacteria that have an outer membrane, but are absent from those that just have one membrane, and therefore that suggests that these proteins have evolved an important function in the outer membrane. Secondly, when you don't have these proteins in bacteria, it often indicates there is an outer membrane defect. So for example here, I have a plate of LB agar, and on the left is the wild type bacteria, and on the right is a triple MC knockout. And what you can see in the presence of detergent, that the triple knockout cannot grow. And sensitivity to detergents often indicates there is an outer membrane defect, because normally this outer membrane would act as a physical barrier to protect the organism from these detergents. Third, these proteins are normally associated with the components of transporters, most commonly ABC transporters, so this suggests that they are transporting something. And then finally, the 3 MC proteins found in E. coli all co-purify with phospholipids attached, indicating that phospholipids are indeed their substrate. So altogether, this tells us that MC proteins are transporters that probably transport phospholipids, and that they have an important role in the bacterial outer membrane. So what our lab wanted to ask was, well, how do MC proteins actually function as lipid transporters? And to do this, I'm going to focus on just one protein found in E. coli. There's 3, but I'll focus on just this one, and that's called LetB. Formerly, it was called YEBT, but I'll get onto why I renamed it, or we renamed it LetB on the next slide. But LetB is the largest MC protein found in E. coli. It has 7 of these MC domains in a row, and it has an N-terminal transmembrane region, which we know allows the protein to be anchored to the inner membrane, with the bulk of it then facing out into the periplasm. What we also know is that LetB is transcribed from the same operon as LetA, which is a predicted transporter protein, and therefore that these two together likely function as a transporter. So for the rest of the talk, I'll just talk about LetB, which is this predicted MC protein, and I'll show you a little bit more about how this may be functioning as part of this transporter. So to start off with, we wanted to find out, well, what does this look like? So therefore we turned to structural biology, specifically cryo-EM. And this cryo-EM structure was solved by Nicola, who's the joint first author with me on this paper. So just to remind you, this is the gene schematic of LetB. It has 7 of these MC domains stacked on top of each other. For our cryo-EM dataset, we removed the N-terminal transmembrane region just to make our life a little bit easier with the sample preparation. So our cryo-EM structure showed us that the protomer kind of looks similar to the gene schematic, in that you have these 7 MC domains stacked on top of each other, and each of these MC domains are very rich in beta sheets. Then what happens is that this protein actually homo-hexamerizes along its length. So 6 of these elongated protomers come together to form a hexamer. If I then, for example, were just to show you this bottom subunit, so this is a subunit made by one of the MC domains, and kind of pull this out and show you a top view, you would see that there are 6 of them that come together like this. One of these MC domains contributed from each protomer, and they come together to form a ring. So because we have 7 of these MC domains in the protein, we actually end up with 7 of these rings stacked on top of each other, which results in a continuous channel that runs through the center of the protein. If we then look at the surface view of our structure, we can see that this internal tunnel is completely shielded. We cannot see it, suggesting that this is a protected environment. So we're starting to imagine this would be a really nice environment for phospholipids to be transported through the center. So just to put it in context of the cell, this protein is anchored to the inner membrane. We know that, and our cryo-EM structure shows us that it's actually a very large tunnel, and it's around 220 angstrom in length. So what's really cool is then this tunnel is potentially large enough to span all the way from the inner membrane across the periplasmic space and potentially interact with the outer membrane. And it's for this reason we renamed this protein lipophilic envelope spanning tunnel, or LET for short. So having obtained this really cool cryo-EM structure, we had a few questions we wanted to ask. The first of which was, is the length of LetB critical for its function? And that's based on the idea that if it really has to span across this periplasm, you'd expect that if you altered its length, the function would be compromised. So this part of the project was initiated by Mark during his rotation in our lab as a PhD student. And as I told you on the previous slide, one ring in the protein is encoded by one MC domain in the gene. So then in theory, we could make shorter versions of this protein by deleting MC domains from the gene, which would result in a deletion of the ring in the final hexameric structure. So that was our plan. We wanted to make a 6, 5, 4, 3, and 2 ring version of this protein by deleting MC domains from the gene. And then with the idea that they remain anchored to the inner membrane, in theory, these would then become further and further away from the outer membrane. However, we actually had 2 questions we had to ask. First of all, can we actually engineer these shorter versions of LetB? Because we can't just assume that we can pull a whole ring out and the protein still actually fold and assemble correctly. And then secondly, if we can then go on and assess whether or not the length of these proteins is important for the function in cells. So to answer the first question, we used negative stain electron microscopy, and this is a 2D class average of the wild type LetB, where you see 7 of these rings stacked on top of each other, so these 7 densities represent those 7 rings of the wild type protein. If we then tried to remove a ring, we were actually successful, and we're really happy to see we can make a 6-ring version, and then similarly we could make a 5, 4, 3, and 2-ring version of this protein. So we were really, I guess, surprised, but also really happy that we could engineer these forms, and having made these shorter tunnels, we could then go on and test their functionality in the cell. So for testing the functionality in cell, we use this genetic phenotype. So what I'm going to do here is compare the growth of a strain of E. coli that has LetB intact on the chromosome, and then a strain where LetB has been removed. So if we compare the growth of these 2 strains in the presence of detergent, you can see in the strain where LetB is absent, there is a growth defect relative to when LetB is present. I can then take wild type LetB in a plasmid and incorporate it back into the LetB knockout strain, and we see that the growth is restored back to the level of the LetB positive strain. So therefore this plasmid successfully complements the phenotype. What I can then do is use this plasmid as a tool to screen those other mutants that I mentioned on a previous slide and see whether or not they're functional in the cell. And that's exactly what I did. And what I found was that a 6 and a 5-ring version could restore growth, and therefore this protein is functional, whereas a 4, 3, and 2-ring version could not. So then to put this back into context of the cell, this kind of tells us that somewhere between 5 and 4 rings, this protein is no longer functional, and that there probably is therefore some correlation between length of the protein and its functionality. And this probably has something to do with decreasing distance from the bacterial outer membrane. Okay, so the next question we wanted to ask was, is the tunnel that's running through the center of this protein important for its function? So first, I'll just show you a little bit more about the tunnel itself. So it's quite hydrophobic. So the regions here colored in gold are hydrophobic patches. And the tunnel itself is made up by these pore lining loops, what we've named pore lining loops, and they are what are colored in red here. So the pore lining loops, there's 6 of them that are contributed from each MCE ring. And because we have these 7 rings stacked on top of each other, we have these 7 sets of rings that all line up, and 7 sets of loops that all line up. And that's what results in this continuous channel that goes through the center of the protein. If we then look at a couple of examples of these pore lining loops, for example, loop 3 and loop 6, the residues colored in red here are those that actually face into the tunnel and are actually contributing to the lining. And as you can see, they are really commonly hydrophobic. And so it really is these loops that are contributing to the hydrophobic environment of the tunnel. So in order to assess whether these loops and hence an intact tunnel is required for function, I mutated all of these different loops. I deleted them individually. And to test whether or not they're functional in the cells, I used the same complementation assay I explained a few slides ago, where you can see again the wild type can complement a LetB knockout strain, whereas just to cut a long story short, all the pore lining loop deletions cannot. So this suggests to us that there is something important going on here, but we couldn't just assume that this was due to functionality of the protein. It could instead also have something to do with stability of the protein when these loops are deleted. So again, we turn to negative stain electron microscopy, and just a reminder, this is what the wild type protein looks like, where you can see these 7 rings clearly stacked on top of each other. If we, for example, looked at one where the second loop is missing, we can see the protein is very stable and happy, but there is a clear hole where this pore lining loop 2 once was. Similarly, we see a hole when we delete pore lining loop 3 just after ring 3. And so despite the fact that the protein itself is happy, there are these clear holes in the side. So then just to put this back into context, normally in the wild type protein, we cannot see this tunnel. It's very continuous and shielded. However, what we think we've done here is effectively punched a hole through the side of the tunnel, and therefore perhaps phospholipids could start leaking out, and therefore we do require a continuous tunnel in order for this transporter to be functional. So then I just want to take it a step further with these pore lining loops. They're actually quite cool in that they are what are actually making up the diameter of this tunnel. So for example, if I take ring 1 and take ring 6 and show you a top view, these are the pore lining loops here facing into the ring. So when Nicola solved this structure, he didn't just solve one structure, he solved several. And what he found is that these rings can actually exist in both open and closed conformations. So here what we have is a movie moving from the open to closed states. And as you can see, as these pore lining loops move, really the diameter of the rings vary hugely. So then we started to ask the question, like, could these movements in some way be gating lipid movement throughout the tunnel? So if I show you, for example, ring 6, which is the most dynamic of these tunnels, when the ring is open, you can clearly see that there is room for a regular E. coli phospholipid or a regular diacyl E. coli phospholipid to pass through the center of this tunnel. However, when it's closed, it looks like it becomes a bit too tight and this phospholipid would no longer be able to pass through. So this suggests to us that indeed, these ring dynamics probably are responsible for gating lipid movement throughout the tunnel, and perhaps it could also be determining which lipids go through based on their size. However, at this time, we had no evidence that phospholipids really do go through this tunnel. It was speculation based on what made sense to us in our minds, and we did not solve any phospholipids densities in our actual structure. So we then wanted to go a step further and kind of get some more concrete evidence that this is the method by which phospholipids are transported, or perhaps consider that they could be going through some other route on the outside or something like that. So to answer this question, I use this unnatural amino acid called BPA, and BPA, when you expose to UV light, will form cross-links with adjacent CH bonds, including those found in phospholipids. So fortunately for us, the Schultz lab at the Scripps Institute developed this tRNA synthetase system, which, when it recognizes the amber stop codon, will, instead of stopping translation, incorporate this unnatural amino acid BPA. What this then means is that we can incorporate this amino acid at any desired location within our protein just using genetics and incorporating the amber stop codon. So that was our plan, was to incorporate BPA at sites within LetB and cross-link any phospholipids in the vicinity. And the way we wanted to detect phospholipids was to grow in the presence of P32, which of course would then be incorporated in the phospholipids themselves. So just to take you through the workflow of the experiment, this is my E. coli expression strain. I co-transform with the tRNA synthetase system that I've showed you here on the left, and also my LetB expression strain. At the time of induction, I add BPA for incorporation into the protein, and I add P32 for incorporation into the phospholipids. I then lyse the cells, cross-link these cells at 365 nanometers, and purify the protein. I then analyze this protein both by SDS page and by exposing to a phosphoscreen to detect any P32 signal that may be there. So within LetB itself, we chose 5 residues within the tunnel and 2 residues outside the tunnel to detect, to see if we could detect any phospholipids at those locations. I also included the wild type version of the protein without any BPA incorporation as a control. So this is the Kamasi gel of my purified proteins and my cross-link samples. And if I take this Kamasi gel and expose it to a phosphoscreen, we can see really clear P32 signals at the sites within the tunnel. However, if we look at sites outside the tunnel or in the wild type control, you can see that this signal is severely diminished. Similarly, if we look at uncross-linked samples, we see that this signal is way down. So what that tells us is that this signal that we see, which is presumably due to phospholipids being cross-linked, is due to these specific locations inside the tunnel. So this was really exciting for us because it really was the first solid piece of evidence that phospholipids are indeed going through the tunnel, and it supported our favorite model. Okay, so then just to put everything in context, I've shown you this really cool envelope spanning tunnel. I've shown you it's made up of these 7 rings, which are dynamic, and I've told you that phospholipids go through the center. Missing pieces of the puzzle are LetA, which is a predicted transporter protein that we think is probably driving transport through the tunnel, although we still don't know that much about that, and we're working hard to try and figure out some more. We also think that there's a missing outer membrane protein that's going to be involved in the outer membrane interactions. One other thing we don't know, and I was intentionally vague about during the talk, was the directionality of transport, and so that's something else we also need to investigate. So this is one of the many mechanisms by which E. coli have evolved to transport phospholipids or other hydrophobic substances across the periplasmic space. Another example is the LPS transporter. So this transports LPS from the inner membrane across the periplasm to the outer membrane. And rather than transporting it in this enclosed environment, it instead goes along this bridge where just the acyl chains, the hydrophobic acyl chains, are actually shielded. Another example are these ferry or shuttle systems where a soluble periplasmic protein can pick up a lipid from one membrane and deliver it across the periplasm to the other membrane. So as you can see, there's quite a variety in the different methods by which bacteria can do this. And this one system here in the inner membrane, or in fact, this whole system is of particular interest to us because it's also a phospholipid transport system. It's also an MCE system. And actually, just to show you at the end of our talk, We've just published on, preprint on bioarchives of the high resolution cryo-EM structure of this ABC transporter. And this is actually, MLAD is a single domain protein, so it just contains one MC domain. So it forms just one ring as part of this bigger ABC transporter. And what's also really cool in the structure is that we found 2 lipids in these unusual conformations. And as you can see here, the densities for these lipids are very clear. And so, yes, we put this on bioarchive just last week, and it actually ended up being one of 4 preprints on this complex last week. So it was kind of a crazy week for us. Okay, so then to finish off, I would like to acknowledge everyone in the Bhabha-Ekiert lab. It's really a great work environment, and in particular, I want to thank my supervisors, Damien and Kira, who they really are amazing mentors, and I consider myself very lucky to have been doing my postdoc in their lab. I also want to thank Nicola, who, as I said, was co-first with me on this paper and really was an amazing person to lead this project with. And like I said, the paper would not exist without his input. I also want to thank Mark for his contribution to the length variance parts during his rotation, and he is now a full PhD student in the lab, also working on these MC systems. I also want to thank our various funding sources, including the AHA, who are funding my postdoc fellowship. And I also want to thank everyone here who came and has listened to my talk today. Thank you very much.
Okay, so there are some questions that have shown up in the chat. I'll let you all chime in and ask your own question. So first one was from Shamshad Cockcroft.
How does the protein, the tunnel, go through your peptidoglycan wall?
So the short answer is we're not sure. But we do observe that there's a kind of gap between rings 4 and 5 here, which we can imagine is where the peptidoglycan may be interacting, and perhaps the spacing between the 2 rings is related to accommodating the peptidoglycan. But how exactly the protein folds, you know, and is inserted through the peptidoglycan, we currently don't know.
Okay, thank you.
Okay, there's another question from Forrest Bowling.
Hi, yeah, that was a great talk. As I was like looking at the loops on your channel, it like kind of reminded me almost of like a crabbing trap, where like the loops are all pointing this like same directionality. So I was wondering, like, you mentioned they have an open and closed conformation. Is like the diameter of the pore in the open state like an equal length from both like perspectives of this hole? Like, could that potentially like force the lipid to travel in one single way and not having like back trafficking?
Yeah, so that's an idea that we have, that it could be, you know, forcing it through and then preventing the backflow, and perhaps the continuous energy, whatever the energy source is, we don't know, could be driving these lipids through. The size of the diameter does vary quite a lot. The ring dynamics also, though, varies in the different rings. So there's probably certain rings that are more like gatekeepers, and probably if you imagine the backflow, preventing the backflow would probably be more important. So ring 6 in particular probably is a gatekeeper for, if there is backflow concern, would be preventing that. But yeah, I think that's totally something that we imagine would be happening.
Thank you.
Okay, John Burke has a question.
Georgia, very nice talk. Very excellent work. Okay, 2 questions, which I know I'm not supposed to ask 2 questions, but I'm organizing, so I'm going to break the rules. First one is, have you played around with changing the size of the pore by making like a subtle mutation on one of those hydrophobic residues to see what the effect is? So, you know, kind of getting at the idea of a lipid specificity, what size lipid do you actually need to allow through the pore? And then the second question is, do you think there's anything that's regulating the opening and closing of the pore?
So to answer your first question, the only way that we've currently altered the pore size is by individually deleting those pore lining loops. We have changed some of the hydrophobic residues to alanines, but in terms of like more like designing experiments specifically to change the pore size, we have not done that yet. But of course, that's something very interesting to see whether it could change lipid specificity. And then what's regulating the open and closing? Yeah, we don't know what's regulating the opening and closing. I would suspect that it could be altered some way in the presence of its inner membrane protein here, but it clearly does this on its own because this is a soluble version of the protein that's not in that we solve the structure of. So it clearly doesn't need to be embedded in the membrane to do that. So, you know, I don't know exactly what could be determining the open and closed states, whether it's something in our, you know, buffers, the presence of lipids or something like that, being different in our different individual, you know, single particles. But it's a good point. We're not sure.
Okay, Tim Levine had a question.
Thank you very much for a great talk. I'm going to change the question from the one I wrote down to asking you if you think this hexameric arrangement and stacking is present in mammalian protein, eukaryote proteins, and not just the ones in chloroplasts. But you know, it's a brilliant system. You know, do you think it couldn't have been inherited from the last common ancestor?
So I think there are other proteins that form kind of rings, but I think the way that the protein is made and that it hexamerizes along its length, I think as far as we're aware, that's quite unusual. We are interested in knowing whether there are homologs of these in mammals or mammalian cells or eukaryotic cells other than the MC systems in chloroplasts. To our knowledge, there aren't, but it might just be that they're so diverged that we can't detect them. But I do think that these systems existed very, very early on in evolution, but you know, evidenced by the fact they're in chloroplasts. And I think they were originally also in mitochondria, but have been lost from mitochondria or drastically changed in mitochondria that we can't even detect them anymore. But yeah, as far as I'm aware, there is not a system that kind of works, or at least you know, comes together in this way, unless anyone else in the audience or knows better than I do.
Cool. So I apologize if I mispronounce this person's name. Diorge or Diorge Souza?
George Souza, no problem. So great talk, congratulations. So the LetB channel has 6 inner membrane helices. So do you know if these helices form a channel, or they're just like anchors, like in the inner membrane? And if they interact with LetA, for example? Yeah. And another question that I would like to ask is, so do you know from where like these lipids come? So are they extracted from the inner membrane? Do you know any mechanism of how this occurs?
So I think both of your questions will be answered once we get the structure of LetA together with LetB. To answer your first question, maybe if I can go back, can I go back a few slides? No. Just that we have like the perfect, okay, well, my computer's not liking what I'm doing right now. So basically, our structure of the MlaFEDB complex I just mentioned a minute ago, here you go. Maybe I can share this again now. We actually got the structure with the transmembrane helices. And yes, so I imagine that what happens in LetB is going to be quite similar to what happens in MlaD, in that you have these 6 transmembrane regions that go around this transporter in the inner membrane. And these do all interact with this, permease subunit here. However, the LetA is quite different to MlaE, so it could be end up being quite different. And then in terms of where the lipids come from, we imagine, for example, if they're extracted from the inner membrane going outwards, then it would be due to some kind of energy source that's transported. This kind of similar system to this would be extracting the lipids. But if it's coming from the outer membrane, then we don't know yet.
Okay, thank you very much.
Yeah, so I think we have to move on, unfortunately. So guess, I'm not sure who's introducing the next speaker, but Georgia will try to answer some of these other questions in the chat.
Perfect. Thank you, Georgia. That was a great talk. And thank you, Damien. All right, so there are a lot of questions that are unanswered, so Georgia will answer those in the chat box, so keep an eye open for your question being answered. And with that, I'll introduce Brian Kobilka, who will introduce John as the next speaker.
Can you hear me? All right. My name is Brian Kobilka. I am a professor at Stanford University, and John Janetzko is one of my postdocs. John has been in the lab a couple of years now. He received his PhD from Harvard in organic chemistry, and he's transitioned to becoming a first-rate membrane protein biochemist and structural biologist. So I will leave it to John.
Great. Well, thank you, Brian, for the introduction. And I'll also follow Georgia's lead. And I want to thank John and Mike for giving me an opportunity to speak in this seminar series. And the project that I'm going to tell you about, which, you know, it's a story about the recognition of GPCRs by beta-arrestins. It's really a GPCR story with a little bit of lipids. And as a feature, since I know you have to sit through to hear the lipids, I put the lipids at the end of the presentation. So I'm excited to give you guys a nice little walkthrough on some interesting GPCR structure and how I think a project that did not originate as a lipid-based project might provide some interesting roles for these lipids in a biological function. And I also want to say that this project could not have happened without, and I'll mention this more at the end of the presentation, but this was really a massive team undertaking with a number of postdocs to achieve this goal. And so since I know this is a pretty general audience, I thought I would just very briefly introduce G-protein coupled receptors, or GPCRs. They're the largest superfamily of transmembrane proteins with about 800 members in humans. And they're important in almost all forms of physiology, if not all forms of physiology, because they're the receptors that are responsive to hormones, neurotransmitters, as well as photons, odorants, etc. And they share this common 7-transmembrane helix fold, 7TM fold. And generally, when people think about G-protein coupled receptor signaling, they think about the action of an agonist at the receptor that leads to an outward movement of TM6, which is the hallmark activation for a family A receptor. And then that initiates the G-protein mediated signals that come downstream, so the engagement with the heteromeric G-proteins. What I'm going to talk to you about today is actually a different process, and that is the process in which G-protein signaling is blocked and receptors become desensitized and a discrete set of intracellular signals become activated. And I'm going to call that the arrestin-mediated signaling process. And in that process, there's two main players. There are these kinases that recognize active forms of the receptor and phosphorylate them. And there are these proteins called arrestins that translocate from the cytoplasm, where they're in an inactive state, to the plasma membrane in recognizing an active and phosphorylated receptor. And in doing so, they block the ability of G proteins to engage the receptor, and in driving internalization of the receptor to endocytic vesicles, they also turn on a set of signals, including ERK and MAP kinases. And what I want to draw your attention to is the structure of these receptors. We've learned a dramatic amount about how G proteins engage receptors over the past few years owing no small part to the advances in cryo-electron microscopy. But in the context of understanding how arrestins recognize receptors, we're still relatively naive. And at the time where we started working on this project, there was no structure of a receptor bound to a beta-arrestin, and I'll clarify that point in a minute. The only structure of an arrestin bound to receptor complex was for rhodopsin bound to visual arrestin, and I'll also go into the details of why we think that warranted further study. So I'm going to broadly break the problem into 4 features. And I'll say that the first feature for obtaining a complex was essentially finding the right receptor, finding the right kinase. We would need to prepare arrestins for this that would engage the receptor. And then we would have to optimize the phosphorylation of the receptor, since we knew that phosphorylation was such an important part in driving complex formation. Optimize complex formation itself, and then ultimately we'd have the task of determining the structure. So I'll introduce arrestin first. So arrestin, in humans, there are 4 arrestin isoforms, 2 visual and 2 non-visual. The visual are specified for rod and cone cells, and then the 2 non-visual arrestins, arrestin 2 and 3, also known as beta-arrestins, they're the arrestins that are responsible for recognizing and desensitizing hundreds of other receptors. And so this already is begging a very interesting question about how do you get specific recognition, but also have the ability to recognize diverse receptor scaffolds. Now, the structure of arrestin basically has two lobes, a C lobe and an N lobe, that are comprising beta-stranded sandwiches. And it exists in an autoinhibited inactive conformation basally. And the inhibitory element is the C-terminal tail of arrestin, which folds back and binds into the N lobe of arrestin. And what was understood is that upon binding of a receptor, these loops in this region that I've called the central crest here, they're going to reorganize their position in order to accommodate the receptor. And the phosphorylated receptor C-terminus is going to displace the C-terminal tail of arrestin and allow that arrestin loop reorganization, as well as other features of arrestin activation. One other feature of arrestin activation is actually an interdomain twisting, whereas if you imagine a screw axis along this plane, the C lobe will rotate relative to the N lobe, and this allows for exposure of other contacts, as you'll see later. Without going into the details of how we arrived at the specific receptor that we were looking at, I'm just going to cut to the chase and say that we had identified the neurotensin type 1 receptor as a particular, the neurotensin type 1 receptor as a good candidate for structural studies because it was able to form a strong interaction with a beta-arrestin. And just the specific details of the neurotensin receptor don't matter particularly for the presentation, other than to say that it's a class A peptide binding GPCR, and its endogenous peptide, the neurotensin peptide, is a 13-mer peptide that binds with high affinity, and it's been implicated in a number of pathologies, including addiction, schizophrenia, cancer. And so with the receptor in hand, the sort of changing gears to the second step in our workflow is to look at receptor phosphorylation. And there's a couple of key features I want you to take away from this presentation. So phosphorylation of the receptor is generally at multiple serine and threonine residues along the receptor C-terminus. And we used an in vitro approach in which we took purified kinase and purified receptor. in order to phosphorylate the receptor. And in order to drive that kinase phosphorylation, we actually have to add an acidic phospholipid. So PIP2, a soluble version of PIP2, is added to the receptor in order to drive that process. And in order to monitor phosphorylation of the receptor in real time and for our structural preparations, we developed a series of ion exchange protocols to basically resolve unphosphorylated from phosphorylated receptor. And so you can see you have discrete separation between those two species. And if you measure a time course, you can see that phosphorylation saturates after about 20 minutes. Now with phosphorylated receptor in hand, we asked a question about arrestin coupling. Sorry, it keeps advancing, unfortunately. We asked a question about arrestin coupling. And so what I've shown here are two size exclusion traces that are comparing unphosphorylated receptor and phosphorylated receptor. And I'm also comparing here full-length arrestin versus an arrestin construct where we've removed that autoinhibitory C-terminal domain. And it was previously known that doing so increases the affinity of the arrestin for the receptor. And what you can see is that unphosphorylated receptor, neither the full-length or the pre-activated arrestin form a stable complex, whereas in the case where you have phosphorylated receptor now, we see that full-length arrestin is able to engage that receptor, although not completely. And if we use a C-terminally truncated construct of arrestin, we can further drive that complex formation. When we looked at our complex by negative stain EM, we were very pleased to find that you can see here arrestin residing underneath the detergent micelle. These are a couple of different views, suggesting that it was engaged with the receptor in a stable fashion, and the sample was quite homogeneous. But unfortunately, it did not survive grid freezing for cryo-EM. And so just to sort of present that in a different way, we can imagine that when arrestin is bound to a receptor, it can be bound through phosphate-specific interactions, but not be fully engaged with the receptor core, which would be in equilibrium with this stable core-engaged bound state. And then both of those species could be in equilibrium with a dissociated or a binding and unbinding state of the arrestin. And in order to facilitate our structural work, we screened a number of additives, complexing conditions, and chemical crosslinkers ultimately, using tools, biophysical tools in the lab of EPR, fluorescence, as well as negative stain imaging to try and optimize this complex. And for sake of time, I'm not going to go into the details, but I wouldn't be giving you this presentation if we hadn't have been successful in obtaining the structure. And so we were able to obtain a structure with a global resolution of about 4 angstroms of beta-arrestin 1 or arrestin 2, and I'll use arrestin 2 for this presentation to not be ambiguous with the visual arrestin, bound to the receptor in a detergent micelle. So here's the micelle shown in gray. And the resolution was sufficient for us to build a molecular model. And I'm going to spend the rest of this presentation sort of taking you through the features of this model. And one of the first things that we noticed when we were analyzing the data was that there was a large amount of conformational heterogeneity observed between the arrestin and the receptor. And one way to probe conformational heterogeneity is a technique called multi-body refinement, where you essentially express a motion as a set of eigenvectors. And what we found was that the first 2 eigenvectors could capture about 40 degrees of the variability in our highest resolution subclass. And you could see that the first eigenvector basically represents a in and out of plane motion of the receptor. Imagine you're looking towards the helix 8 of the receptor. And the second eigenvector was a rotational motion in which the angle of arrestin is changing relative to the receptor. And yeah, if we look at the receptor itself, what we see is that the receptor exhibited the canonical hallmarks of activation for this class A receptor. So again, TM6 is extended about 11 angstroms outwards from the inactive position. As well, there's an extension of TM5. And if we compare it to a previously solved G protein structure bound to the neurotensin receptor, we see that the G protein bound state of the receptor and the arrestin bound state of the receptor are quite similar. When we look at arrestin itself, we sort of see those features that I had mentioned to you in the slide where I introduced arrestin. And so if you compare the wheat sheet here to the blue sheet, which is in the arrestin-bound structure, you can see that there is a twist of these beta strands about 16 degrees relative to the fixed N-terminal domain. You can also see a reorientation of these loops in the central crest. Specifically, I'm going to focus on the finger loop, which is now extended and has a helical property to it, as well as this gate loop, which has gone from a folded in conformation to now an outward extended conformation. And what really surprised us when we compared this to the previous structure of rhodopsin bound to visual arrestin is that the orientation that arrestin assumes when bound to the receptor is dramatically quite different. It's rotated about 90 degrees relative between the two structures. And what that means in practice is that if you look at the interface between the arrestin and the receptor, the same loops are making contacts and holding the receptor in place. However, the points on the receptor that they contact are different in between these two structures. And what was satisfying for us was that if you look at ICL2 specifically, which is forming a strong close contact with the C-loop of arrestin, is that some of the residues that are most highly conserved for arrestin recruitment in ICL2, specifically this proline here, which is residue 173 in neurotensin, is right here and orients a hydrophobic residue in close proximity to the C-loop, sort of affirming the importance of this interaction in the complex. When we compared the neurotensin structure bound to arrestin versus bound to G protein, we see that the projection of the arrestin finger loop is along the same trajectory that the alpha-5 helix of the G-alpha-I subunit takes. However, the finger loop doesn't extend as deeply into the receptor lumen as the helix, suggesting that this may allow for more conformational flexibility within arrestin. If we look at the phosphate-specific contacts, again, I had mentioned before that the gate loop is now in an extended conformation. We can see that it's now moved proximal to a phosphate residue in the receptor C-terminus. And sort of, it was known that this is an interaction that is particularly important for mediating arrestin activation. And we also saw that an extension of ICL3, or a region of ICL3 extending out from transmembrane helix 6, was further ordered than previously seen and ended at where a residue that we saw was phosphorylated, serine 287, would be brought proximal to the base of the finger loop of arrestin, and specifically two positively charged residues, lysine 76 and lysine 70, arginine 76 and lysine 77, residues that an interaction which may stabilize the extended conformation of the finger loop in the structure and further rigidify this complex. Now, I know this is a lipid-centric audience, and so I would be remiss to not discuss the story of how we identified the lipid bound to our complex. And what I'll say is that at one of our earlier maps, we originally had this somewhat mysterious density that wasn't explained by the receptor of the arrestin. And we wondered whether this was an inositol phosphate that was bound. It had previously been shown that the C-terminal lobe of arrestin can bind inositol phosphates. And some native mass spectrometry work from the Robinson lab had also suggested that inositol phosphates might bridge between the transmembrane regions of receptors and potentially transducer proteins. And so I'll cut to the chase and just say that when we took a cryo-EM sample where we did not add exogenous inositol phosphate, soluble PIP2, into our sample, we could actually detect that there was indeed inositol phosphate that was co-purifying with the receptor. And this is pretty remarkable because if you remember from the workflow, we're adding that PIP2 at the stage of promoting receptor phosphorylation, which has many steps of chromatography upstream before finally our cryo-EM analysis, suggesting that the interaction must be pretty tight. And so we felt confident to model PIP2 into that density. And what we see is that the phosphates of PIP2 are making contacts to a number of positive charges in that arrestin C-lobe, as well as contacts to transmembrane 1 and 4 of the neurotensin receptor. We then directly measured binding of a fluorescent derivative of PIP2 to both the neurotensin receptor and to arrestin, and found that it bound with extremely high affinity in both cases. And if we made a mutation where we removed 3 of those positive charged residues, we would dramatically reduce the affinity of the PIP2 for the arrestin. And taking that one step further, we tested that same mutant in the context of cells using a split luciferase assay, where we look for complementation or proximity between the receptor and arrestin. And we found that in the case where you mutate those 3 positive charges again in the C-lobe of arrestin, you would significantly reduce the amount of arrestin that was recruited to the neurotensin receptor. Now, one final thought I want to leave you with is something else that stood out to us, and that was when we looked at our complex, we asked, why is it that arrestin is potentially so tilted relative to other transducer complexes? As you can see here, this region of what is called the C edge of arrestin, which is known to interact with membranes, would actually have multiple residues completely buried inside the membrane in the conformation that we observed in our structure. Compared to a G protein. These are sort of simulated membrane demarcations. But one thing that we started to think about is that membrane environments are not planar, and you can imagine that as you increase the degree of membrane curvature, that might account for the fact that now you would need fewer of those residues to interact with the membrane. And so perhaps what we're seeing, although in part being driven by the fact that we have a small detergent micelle that's not exerting any downward force to push the arrestin into a more planar conformation, but perhaps this state is relevant in the context of when this complex has to co-traffic into regions of higher membrane curvature, arrestin might be able to deform and change shape in order to accommodate that curvature. And so with that, I want to leave you with a couple of key takeaways. So we were able to obtain the first complex of a receptor bound to beta-arrestin. And in doing so, we learned about the high degree of conformational flexibility exhibited in the arrestin. We also saw key examples of phosphate-specific contacts between both the C-terminus of the receptor and the third intracellular loop, and also learned about potentially these lipid interactions that mediate bridging arrestin to the receptor and might fine-tune the interactions. And so with that, I want to thank all of the people who did this. I did not do all of this work. And so this was, as I mentioned at the beginning, a team effort between four postdocs. And Matt, Vincent, and Q and I worked to make this project happen. I want to thank Brian and Yurgo for their mentorship and sort of leading the charge on all of this. Our collaborators, Jeffrey Gunn's lab that helped with PIP2 analysis, Asuka Inoue for cellular assays, some folks at Brooker who gave instrument time, And then I want to thank the funding support, Damon Runyon Foundation for funding me, NIH for funding the lab, and then Brooker and Slack for instrument time and access. And I want to thank you all for your attention.
All right, John, very cool talk. Brian, do you want to field questions?
I think John can field the questions.
Okay, yeah, John.
I'll be available.
Hi, can you hear me?
Yep.
Yes.
Hi, so I'm sorry for, this is Anirban from the NIH. I'm sorry for asking sort of a in the weeds question, but my question was that for the complex, the optimal detergent, do you need to use a detergent with a longer alkyl chain to keep the membrane binding surface of the arrestin happy? In other words, does the detergent micelle need to sort of reach out to the surface between the GPCR and the arrestin to keep the complex happy as compared to the optimal detergent for the receptor only? I don't know if the question makes sense.
Sort of, yeah. We used, so this is an LMNG micelle, and I guess it was, so there was another paper that was published at the same time that looked at a muscarinic M2 receptor in a nanodisc, and they found that the size of the nanodisc, so the nature of the lipidic environment mattered, where you needed to have enough lipid in order to satisfy this arrestin, in order to make contacts in this region here. And so I guess, I don't know if that's answering your question directly.
Yeah, I mean, I was wondering if you know what's the optimal, what's the shortest detergent in which NTSR1 just in itself is stable. And you may not actually have done that experiment on your own if you were just going for the complex. So, if not, then don't worry about it.
I can answer that for GPCRs in general. So we've done this with a lot of different GPCRs, and generally anything shorter than 12 carbons are destabilizing. LMNG is a special design detergent. It's actually two detergent molecules bound together, but it has about the same total length between the sugar and the last carbon of the alkyl chain. So then, you know, it's really necessary for the receptor. And we haven't tried anything longer, but this range seems to be optimal for most GPCRs.
Thank you.
John, you had a question?
Yep. I'm just kind of big picture question. So in theory, you know, arrestin signaling at the plasma membrane and in the endosome compartments, you would think as the endosome compartments are going to be getting more and more depleted in PIP2. So I guess my question is, A, do you think this is a mechanism of how arrestin signaling actually gets turned off as endosomes lose PIP2? And secondary question, do you think there's any other possible phosphoinositide that could compensate for that interaction? Have you looked at that?
Totally, yeah, that's an awesome question. And I will say that there is some ongoing work that I've been doing to sort of try and understand that process and tease that apart. What I can say is that at least broad strokes, there seem to be, so what was known for a while is that different inositol phosphates bind with a different affinity to arrestin. So if you compare PI3P versus PIP2, you have a pretty dramatic reduction in the amount of affinity that's able to drive. And so it may be, and that's one of the things that we've been thinking about, that as you switch over and you deplete the PIP2, that is a way of sort of promoting arrestin to leave in some of these complexes. And what was the other part of your question?
The second part was, well, I mean, same thing. So just essentially that as you lose PIP2 on the endosome, it's a way of turning off arrestin signaling.
Right. Oh, you also asked about generality. Yeah, and so what I can also say is that we see for that this is not uniquely a neurotensin case, and that there are a number of receptors that exhibit similar behaviors, and that receptors are either very sensitive to or relatively insensitive to this inositol phosphate. And we think that we might have an idea as to what sort of drives that. And that's very much ongoing work, and I think would really tie together that generality. Josh, you had a question, Joshua Pemberton?
Yeah, great talk, John. I guess it's a little bit similar to John's question in terms of, you know, if you look at neurotensin as a model, you have Carol Robinson's group showing that PIP2 regulates the recruitment of the G protein heterotrimer, maybe even the GTPase activity. You also have PIP2 regulating GRK recruitment to the membrane through these polybasic domains and phosphorylating the receptor. Now you have PIP2 as a stable associate for the arrestin recruitment, but the neurotensin receptor also couples to G-alpha-Q. So if it's running the PLC system in the plasma membrane, what's happening to these recruitments as you deplete PIP2 in the membrane and you start having to turn over the PIP2? So all of these features are seemingly happening at the same time. And how do you have this stable recruitment of a PIP2 molecule when it needs to be passed through all these different conformational states of the receptor?
Yeah, I think that's a fantastic question, and very much, I don't think that I have a perfect or a particularly great answer for you.
No, I don't expect you to, but I just have
yeah,
started
but,
to speculate. Yeah.
Yeah, so I guess there's a couple of things. So one possibility is there may be a relative difference in kinetics between PIK, recruitment of arrestin, recruitment of GRK, and PLC activation. So it's been shown for like GERK activation, for example, that not every receptor that activates and produces G beta gamma is triggering GERK activation, and that a determinant of that may be coming from just rate and turnover numbers. So that's one possibility. And another is that, as John's question sort of alluded to, is that you really have an issue of you don't want signaling from every subcellular compartment. And so some of these steps you might want to confine into different regions. What I can say as a third point, and it's not something that's unknown, but so when I had discussed the cellular data that shows that neurotensin, that you need to be able to bind PIP2, or you have a reduction in the amount of arrestin that gets recruited to the neurotensin receptor when you don't have the same amount of PIP2 binding capacity in arrestin. There are receptors for which they are much more sensitive to this process than the neurotensin. So even though we have a reduction that we see, other groups have shown that for something like the beta-2 receptor, beta-2 adrenergic receptor, you completely lose the ability to recruit arrestin if you can't bind PIP2. And that may be because the neurotensin, as you said, it's activating GQ, it's going to deplete PIP2. It can't be exclusively dependent on PIP2 for that interaction. Otherwise, it won't be able to get deactivated.
Yeah, so just one really quick follow-up. With Mark von Zastro's group with the 3Q mutant and the C edge, so then do you think the C edge of arrestin is binding first? Because this is where, when it associates with this PIP2 molecule, the communication between the C edge loop and this little pocket is not really clear to me from some of these structures. I just didn't know if you had any ideas about that either.
No, at this point, I don't think we can really comment on the C edge and particularly on the order of events. It's reasonably poorly resolved in the structure. It's kind of one of the areas of lowest quality density, presumably because it's mixed in with the detergent micelle. yeah, I can't really speak to the order of events.
Very cool. Thanks very much.
For sure. Mary, you had a question? Mary Roberts? Maybe it.
What is very neat about the structure is the orientation of your arrestin with respect to the receptor. And you have this lovely curved micelle. You said people have done it in nanodiscs. Have you tried bicells? Something where you could have a flat surface to reconstitute and to see what it looks like?
So I can say that we have tried bicells. There is no, to my knowledge, there's no cryo-EM structures that have been determined in bicells.
Okay.
We weren't, previous work before my time had been successful with crystallographic efforts at this complex. And so the other thing with bicells is that they tended to be a little bit more finicky than nanodiscs. And so we had tried things in nanodiscs. And if you look, the structure that was solved in the context of a nanodisc, there's a lower degree of curvature for the arrestin because it is, again, a more flattened membrane.
Okay.
It's not perfectly, but I don't know if that answers your question.
It does. Thank you.
Okay. And then Mike, you had a question?
Yeah, John, fantastic talk. I think you've sort of alluded to this already, but my question was, what sort of features are specific for this system versus what features do you think may be generally applicable to other GPR systems or particular classes?
Yeah, so I guess broadly, I think that every receptor will have engagement through the receptor lumen. I think there's a number of examples where the vast majority of receptors seem to have this phosphorylation-dependent recruitment of arrestin. And so I think that the phosphate-specific contacts are going to be conserved. And there's been a couple of computational papers now that have looked at a number of receptors and their ability to concentrate PIP2 and acidic phospholipids at various points in their transmembrane region. And what is pretty striking is that if you compare something like neurotensin to something like the dopamine D1 receptor, I believe, they find that there's different regions along the helical bundle that seems to concentrate these PIP2 lipids, suggesting that you might have a pretty wide range of different positions, and that the position of the lipid might vary to sort of synergize with that phosphate-specific contact. So neurotensin receptor is understood to predominantly couple through its, engage arrestin through its C-terminal phosphorylation. But for some receptors, they have very short C-termini and very large intracellular loops. And if that loop is where all the phosphorylation sits, you might want to rotate the complex because now you need to be able to accommodate that loop sitting in that phosphate binding groove. And so it may be a fine-tuning element where it moves around the receptor depending on where the phosphorylation is.
Thank you.
Brooke Gardner? She's still there. Maybe.
Can you hear me?
Oh, yes, hi.
Hi. So my question was if you think that the conformational flexibility that you're seeing is a feature of the function of the system, or is it because you think you're missing some sort of stabilizing binding partner in your structures?
So that's a good point. So I think that it's probably or possibly a little bit of both. I think that in the case where you have a detergent micelle that's pretty small, you can have a little bit more motion along that micelle. This contact through the C edge is relatively minor, and if you imagine this PIP2 molecule, if it can come out or deeper into the micelle, it can change the angle in which the arrestin is bound. The other thing is, and then it wouldn't be directional in the in and out of plane motion. The other thing is that we know that our receptor phosphorylation is not perfectly homogeneous. And it's also possible that if the receptor is bound through predominantly different phosphates, that you're looking at an ensemble of poses. And we would expect that that would be the same case in cells. It's known that phosphorylation of these receptors is pretty diverse in cells. And so it may be that you have multiple different complexes in terms of what phosphates are mediating the interactions with the, between the receptor and the arrestin. And so they may be a conformational ensemble in cells as well. If that answers your question, maybe. And then Ray Blind had a question about PIP2 and IP6.
Yeah, I'm just wondering if the PIP2 site is similar to the IP6 site that people have seen in the arrestin-2 crystal structure.
Yeah. Yes. So it is.
You may have said it, and I may have missed it. I'm sorry if I did.
No, that's totally fine. Yes. So I think it is and it isn't. So there's a couple of papers that have looked at arrestin by crystallography with IP6. And if you look in the literature, they suggest that there's a high and low affinity site. Likely the low affinity site correlates with competition and phosphate-specific interactions in the end lobe of arrestin, whereas this high affinity site seems to be both here in the C lobe, and then there's also sort of outside of the C edge in what sort of mediates the arrestin-arrestin multimerization. So it is one of the positions where IP6 was bound, yes.
So do you have any inkling or maybe some insight into whether or not those IP6 or inositol phosphate sites are real, or if they are just a product of phosphatidylinositol binding sites?
I'm not sure I understand the question.
Well, I mean, I guess the idea might be that the inositol phosphates, the soluble, are co-crystallizing, and they're obviously really there, but they may not play a physiological role, and they're just mimicking the binding site for a lipid that has an inositol phosphate group on it.
Yes, it's possible. It's possible that I think it's hard in all of these in vitro systems to fully understand the context of some of these things. And there's the same caveats when you're looking at a cryo-EM structure as with an X-ray structure. So if you see oligomerization mediated by PIP2 at the outset of it, you know, it is possible for it to be bound there, but as you said, it could have, it could just be from elsewhere in a membrane, making a contact there. I think that, you know, this structure gives us a pretty good insight into at least that this proximal PIP2 binding might be mediating directly the interaction between the receptor and the transducer. And beyond the additional sites, I think that it remains to be seen whether those end up having a physiological driving factor. And then Stefan, Stefan.
Hi. Yeah, it's my understanding that this is just a comment related to Josh's question, that PLC only hydrolyzes a small pool of PIP2 under physiological conditions. So PIP2 wouldn't become limiting, at least in the case for receptors that are coupled to GQ. But I was also wondering if you'd examined arrestin localization in cells that are lacking synaptogenin 5-phosphatase activity. Does it become trapped at endosomes?
No, we have not looked at that. Short answer to your question, although I think that's a good idea. I think that's that would absolutely be an interesting experiment to test, and very, very doable.
All right, let's thank John and Georgia for a great set of talks. We will be back next week with another set of talks from the Voorhees lab, and it should be great. So thanks again, and these will be posted online on the ASB website in a couple weeks. Thank you.